This is the pre-lab video for the Macroevolution Lab for Biology 1441. By now, you're probably familiar with Linnaeus's classification system from domain through species, and you can apply it to a number of organisms, including our own mascot. Here's a black bear. We don't have a purple one, sorry. Domain eukaryote, so it has cells with nuclei, right? Kingdom Animalia, so it's within the animal kingdom. It's a multicellular animal. Phylum Chordata, it has a spinal cord. Class Mammalia, so it's got mammary glands. Order Carnivora, family Ursidae, genus Ursus, species Ursus americanus. So when you're writing a scientific name, the genus is always capitalized, the specific epithet, which is this second part, is lowercase, and the whole thing is italicized. Okay? So genus Ursus, the species is Ursus americanus. Now, after Linnaeus's system was devised, we figured out this whole evolution by natural selection concept, and so we now have to mesh the two ideas together. And we do that through the use of cladistics and cladograms. When we draw these trees, these are like family trees that show evolutionary relationships. And any level of Linnaeus's taxonomy can be called a taxon, whether it's a kingdom, family, genus, or whatever. So here's taxon A, B, and C. And on this particular cladogram, we can see that taxon C is the outgroup, meaning it's the first to diverge on the tree. Taxon A and B share a recent common ancestor, which is, we don't know all the ancestors. All of the ancestors that would be down here are extinct. We might have some fossils that we think fit on this tree, but we know the living ones, and that's usually who are listed on the top of the tree. Down here is the hypothetical ancestor of those two organisms. And then down here at the very bottom is the hypothetical ancestor of everyone on the tree at this node down here. So taxon A and B are said to be sister groups because they share a recent common ancestor. So what we do when we build these cladograms are to group organisms into a clade, which is an ancestor and all of its descendants. So for example, chordates are a clade, mammals are a clade. And what we found out, and what we still find out to this day, is when we start analyzing things from an evolutionary perspective, is that sometimes we have things classified into the wrong categories. Something that we thought fit into one category really fits into another. Okay, now, here's some examples of ways we might screw this up. First of all, the top two are good. An ancestor and all of its descendants as a clade. So maybe this is the mammals, right? Or maybe over here you have the birds. Let's just say these are birds on this side of the tree. And here is an ancestor and all of its descendants. Maybe this is a specific family of birds or a specific genus of birds, right? That's a clade within a clade, perhaps. Now, the problem arises when you name, like, let's say these are the snakes over here, and then there's this other organism that you thought was a snake, but it's really a lizard with no legs, and it actually falls on a different part of the tree. And you called this a snake, and you called these a snake, these snakes, but none of these other ancestors below are included. That would not be a clade. Or if you leave out one descendant. So why are these not clades? In this example, you have descendants but no common ancestor included in that clade. And in this one, you've left out a descendant. You have an ancestor and most of its descendants, but not all of them. So sometimes we realize we've made these mistakes and we have to reclassify organisms based on our errors, our earlier misunderstandings of evolution or of relationships. So here's an example of a cladogram, and when we look at these, we often refer to ancestral or basal traits, which are old character traits, and derived traits, which are traits that have more recently arisen and may separate one group from another. And we always have an outgroup at the base of the tree, which roots that tree. It gives us directionality. The outgroup is usually the most distantly related member to everyone else on that tree. And so in this example, we have several clades. For example, everyone on the cladogram we can call a vertebrate. And then within those vertebrates, we have another clade, which includes the last common ancestor of vertebrates that had jaws and all of its descendants. So those are known as the gnathostomes, all of those organisms that have jaws. And then we have the last common ancestor of vertebrates with a bony skeleton and all of its descendants. So the bony fish, which actually includes human beings, amphibians, lungfish, all of those things are really bony fish. And then within that group, we have the last common ancestor of land vertebrates and lungfish, and then the last common ancestor of land vertebrates. So clades can nest within each other. So you could have land animals, which fit into a larger clade, which are gnathostomes, which are organisms with jaws, which fit into a larger clade, which are the vertebrates. And so these kind of mash up with the Linnaean classification system. Here's an example of one of those situations where we've historically had a misclassification. For example, this circle includes what we now know to be the reptiles, an ancestor and all of its descendants. But historically, we've left birds off of that cladogram and have not called them reptiles. But in all reality, we can't exclude birds from the list of reptiles. They share a common ancestor with crocodiles. Now that might seem really weird. Birds and crocodiles don't look anything alike. But remember all of the extinct diversity that is not represented by the living things. All of the dinosaurs were between crocodiles and birds, for example. So when we're making cladograms, we usually use shared characteristics. So you may have heard of the term homologous traits. We're looking for homologous traits that link organisms together. For example, here's a list of organisms: human, bird, salamander, fish, shark. So what's a ancestral or basal trait that humans, birds, salamanders, fish, and shark have in common? They all have vertebrae. Now, a derived trait would be a trait that separates these organisms from each other. For example, feathers is a trait only found in birds. Feathers are a derived trait of the birds. So we use these different kinds of traits to try to figure out how organisms are interrelated. Now, we have to be careful when we're doing that, however, because sometimes we can be mistaken. For example, analogous traits can give us the wrong impression. Analogous traits are similar structures that evolve in different organisms that look similar, but they have different evolutionary histories. For example, the eyes of chordates, like ourselves, and the eyes of certain mollusks, like this octopus, are very similar in structure. And we might easily think that they are shared due to a common ancestor, which would mean we might think they're homologous. But if we look at a cladogram, we can see mollusks are way down here, chordates are way up here. So if that kind of eye evolved in a common ancestor between both of us, we would expect to see it in all of these other organisms as well, but we do not. So in fact, this is an analogous structure, a structure that's evolved due to convergent evolution. So this organism was classified by Linnaeus as a snake, and you might look at it and come to a very similar conclusion. We need to look at its characteristics. And if we build a data matrix, which is the name of those tables where we classify what traits the organisms have, let's have a look. Here's the data matrix. Here's salamander as our outgroup, and then we have lizards, snakes, and this new organism on the list, and we can now try to compare and contrast it with these other organisms. Salamanders do not have an amnion. Okay, that's a membrane in the egg, but these all do. So these are all reptiles in this case. Legs. Salamanders have legs, lizards have legs, snakes do not. This organism does not. So okay, there's why we might call it a snake, right? But let's keep looking. External ear openings. Salamanders don't have that, but lizards do. Snakes do not, but this organism does. That's weird. Unhingeable jaw. Salamanders don't have it, lizards don't have it, snakes do. This organism does not. What about eyelids? Lizards and salamanders have it, snakes don't. This organism does. So when you put all this together and try to make sense of that on an evolutionary tree, you come to realize that in fact that is a legless lizard. It has a derived trait, a recently evolved trait, which is the loss of legs. But that doesn't make it into a snake. Snakes are separated by lizards by millions and millions of years of evolution. This is a lizard without legs. Anyway, we're going to be making cladograms in class and working on that. So review that in your book, and we'll have a good lab this week.